Welcome students. My name is Miss Karen Lenisha Cabral, Assistant Professor in Botany at St. Joseph Vaz College, Kottali, Goa. Today's topic is for Bachelors of Science, second year, subject Botany, semester 4, course code BOC 104, course title Plant Physiology. Title of the unit: Plants Response to Light and Temperature. Module name: Phytochrome, that is the discovery and structure. Outline: Introduction, discovery of phytochrome, structure of phytochrome, functions of phytochrome. Learning outcomes: states the history of phytochrome, describes the structure of phytochrome, and lists the function of phytochrome. Coming to the introduction, when we say phytochrome, what exactly are phytochromes? Now, these are class of photoreceptors which are found in plants, in bacteria and fungi, which it is used to detect light. Now, these phytochromes are sensitive to light in two regions, that is the red and the far red region of the visible spectrum, or when we say the electromagnetic spectrum, we consider these two regions because when we say red light, we talk about 660 nanometers, and far red light comes around 730 nanometers. So, in these two regions, they will either inhibit or they will stimulate growth in a particular plant. Now, according to that, they can be classified as type 1, in which are activated by far red light, or type 2 that are activated by red light. Now, recent advances have suggested that phytochromes also act as temperature sensors, that is, in warmer temperatures, warmer temperature enhances their deactivation. So, they require a particular temperature wherein they will remain in its active phase, but the moment the temperature gets warmer, it will be deactivated. Now, all of these factors contribute to plants' ability to germinate. Other plant photoreceptors include cryptochromes and phototropins. Now, these are again the other receptors that respond to blue light, which respond to blue light and ultraviolet A light, and UVR8, which is sensitive to ultraviolet B light. Now, in addition to their role in nature, the light-induced interaction between a plant phytochrome and phytochrome interacting factors was used in 2002 to control gene transcription in yeast. Coming to the discovery of phytochrome, now in 1932 there were a few scientists who discovered this substance, but they didn't know what exactly it is at the initial stage. So in 1932, Bortwick and Hendrick showed that red light elicits germination of lettuce seeds, whereas far red light inhibited this process. So, when these seeds were exposed to red light, they germinated very well, but when it was exposed to far red light, this germination or their growth was inhibited. Now, when lettuce seeds were exposed to alternating red and far red light, because after they discovered this, what they did was they exposed these seeds to alternating cycle of this, and what they discovered was whenever the seed received red light as the final treatment or the last light treatment, they germinated, while those seeds which received the far red light did not germinate. So, which means the far red light inhibited the growth. Now, this was in case of lettuce seeds. But later on in 1952, when they studied Bortwick, Hendrick and Parker demonstrated that red light inhibited the flowering in xanthium, but this effect could be reversed with the exposure to far red light. In the previous one, red light stimulated the growth of the germination of seeds of lettuce, but here what it did in xanthium, it inhibited the growth of this particular or the flowering of this particular plant, but far red light stimulated the flowering of this plant. The action spectra for inhibition and promotion of flowering shows that the red light near 660 and far red light near 730 nanometer respectively are maximally effective. So each one had their own role to play of either inhibiting or promoting flowering. Now when plant is subjected to several consecutive radiation with red and far red light in sequence, the flowering response is determined by the wavelength of the final exposure. Just as I mentioned before, the last exposure of the light makes the difference if whether the plant is going to flower or it's going to germinate or no. Bortwick and his associates concluded that phytochrome exists as two interconvertible forms, one absorbing red radiation, that is given as PR or P660, and the other with an absorption maximum in the far red region of the spectrum. So if you see over here, they both are interconvertible. So if you see during darkness, the red light PR form will be in its inactive phase and PFR form will be in its active phase. That will lead to cellular response. The PR form of pigment is converted by red light to the PFR form. So if you see over here, the PR form is converted to PFR form in the presence of red light. So conversely, far red light changes the PFR form of the pigment to PR. So in 1959, Butler and his associates first extracted phytochrome from the ethylated oat calyptiles. Now these are seeds which respond or which germinate in darkness. It occurred as a chromoprotein in which the chromophore is a linear tetrapyrrole similar to C-phycocyanin. Now, most studies with phytochrome have used ethylated seedlings from which phytochrome has been obtained in highly purified form. Coming to the structure of phytochrome, phytochrome is a soluble chromoprotein, which means that protein which imparts color, with a molecular mass of around 250 kilodalton. Now it occurs as a dimer, that means it has 2 units and the weight of it is distributed equally, wherein each one will have 125 kilodalton. Now each unit has 2 groups. So it consists of 2 components. One is the chromophore and the other one is the apoprotein. So the light absorbing pigment molecule, that is the first part is the chromophore part and the other one is the polypeptide chain, that is the apoprotein. Now these 2 components together make up for the holochrome. The chromophore is a linear tetrapyrrole which is similar to phycocyanin, termed as phytochromobilin, and it is a ring which is attached to a protein through thioether linkage to a cysteine residue. Now the absorption of red light provides the energy which is required to overcome a high activation energy for rotation around the double bond. Now there is further evidence that the protein also undergoes photochemically induced conformational changes. Now if you look at the structure of phytochrome, it has these 2 identical subunits, that is 1 and 2. They appear in the same way. Okay, now these 2 subunits, if you take just one subunit, it has 2 parts, or you can say it has 2 domains. One is the photoreceptor activity, wherein it will react to the light, and secondly is the kinase activity. So basically kinase activity plays a role with the transfer in the phosphate group. Now in the first domain, that is a photoreceptor activity, now it has this activity because of this group that is present, that is the chromophore group. So phytochrome has 2 different chemical structures that are interconvertible, as mentioned before, that is the PR form and the PFR form. So the forms are named by the color of light that they absorb. Now PR is a blue form which absorbs red light, that is in 660 nanometer, and PFR is a blue-green form that absorbs far red light, and that is in 730 nanometers. Now when they do absorb these photons, they change chemically into the other form. Now both have some absorption in the blue end of the spectrum. So if you see over here, the red light will be somewhere over here, whereas the far red light has somewhere over here, the peak is somewhere over here. So this is the electromagnetic spectrum as per where exactly the far red light and the red light is going to have the maximum value. Coming to the functions of phytochrome. Phytochrome play a role in germination of some seeds. Now it also plays a role in developmental phenomena like photomorphogenesis, that is those plants which develop with the help of light signals, then photoperiodism and clastogamy. It also helps in formation of rhizomes and bulbs. It plays a role in seed dormancy, seed leaf abscission, and synthesis of gibberellin, anthocyanins, carotenoids, etc. That is with regard to the pigments. Phytochrome A and B have distinct role in the regulation of plant growth and development. These are the references. Thank you.